fact behind Mendel's success was the genes encoding his selected traits did not reside in the same chromosome. If they had been, his dihybrid cross results would have been much more confusing, and he would not have discovered the law of independent assortment. Sounds interesting? Let's find that out. Warm greetings, everyone. Today, we'll be looking at genetic linkage and recombination. We'll be going through a short introduction, then the definition of genetic linkage and recombination. In a pair of chromosomes, there are several number of genes. A theory put forth by Walter S. Sutton and Theodore Boveri states that chromosomes are the physical basis of heredity, carrying the hereditary factors or genes. This theory is called the chromosome theory of heredity. Volta suggested that these genes do not undergo random assortment as proposed by Mendel, but rather they are inherited together during the process of inheritance from one generation to the next. So what is Mendel's law of independent assortment? During meiosis, the chromosomes in each pair, they separate one to each gamete, or we can say that the pairs of chromosomes, they assort independently of each other. But here Walter says that they are inherited together during the process of inheritance. Keeping that in mind, let's go to our next topic, which is genetic linkage and recombination. The term linkage was coined by Thomas Hunt Morgan. He is known as the father of experimental genetics. It was he and his colleagues who conducted several experiments and gave confirmation to the chromosome theory of heredity. Now actually, what is linkage? The genes in a chromosome are linked together and they belong to the same linkage group. So, the tendency of the DNA sequences that are close together on the chromosome to be inherited together during the process of meiosis of sexual reproduction is linkage. Or we can say that it is the tendency of two or more genes of the same chromosome to be inherited together during the process of inheritance. If there is any alteration or mutation, which is the abrupt changes to the DNA, it will result in genetic recombination. Now, what is genetic recombination? Before moving on to the definition, let's consider a pair of homologous chromosomes. Homologous chromosomes are those chromosomes which are having equal gene sequence, same loci, chromosomal length, centromere location, and they belong one from the paternal and the other from the maternal, and they pair up during the process of fertilization. So what happens here is that these homologous chromosomes, they undergo reassortment or crossing over, which is the exchange of genetic material, and they result in the recombinant chromatids. So we can say that recombination is the formation of new genotypes by reassortment or crossing over of genes, and there is exchange of genetic material between two homologous chromosomes. So, we have discussed two topics, which is the linkage and recombination. So, hope it's clear to everyone what we have discussed today. Thank you all for your patient listening, and thank you all once again. Have a good day. Thank you.